Hello, everyone. Welcome to the fourth installment of the OpenKMI Modeling Initiative's quarterly webinar series. Today, we'll have a seminar by Julie Price from Harvard Medical School and Massachusetts General Hospital. After Julie's presentation, she'll have a panel discussion with our panelist, Bob Koeppe from the University of Michigan. We'll also invite you, the members of the audience, to submit your questions and participate in the discussions interactively. My name is Marc Normandin, and I'll be the moderator of this session. That means that I have the honor of introducing today's speaker, Dr. Julie Price. She's a professor of radiology at Massachusetts General Hospital and Harvard Medical School with expertise in translational PET imaging of protein targets in aging and neurodegeneration. Julie completed her bachelor's degree in physics and master's degree in medical physics at the University of Wisconsin at Madison, earned a PhD at Johns Hopkins University, and completed postdoctoral training at the NIH Nuclear Medicine Department. Dr. Price was on the faculty at the University of Pittsburgh for many years, where she helped to establish PET imaging of amyloid plaque for human use. In 2016, she joined Harvard and MGH, where she's the director of PET pharmacokinetic modeling at the Martino Center for Biomedical Imaging and faculty co-director for research at the MGH Center for Diversity and Inclusion. She currently holds R01 research support and is director of the T32 training program from NIH. Dr. Price's achievements have been recognized through many honors, including induction as a fellow of the Society of Nuclear Medicine and Molecular Imaging, and receipt of the SNMMI Brain Imaging Council's Cool Lassen Award. Today, we're fortunate that she'll be sharing her lecture entitled Kinetic Modeling of Neuropathological Protein Accumulation. Julie, the floor is all yours. And I think you're on mute.
Good morning, everyone. I'll talk for just about 40 minutes, and I'll be telling a story across time about kinetic modeling of neuropathological protein accumulation. I'll start by defining the hallmarks of Alzheimer's disease, which are amyloid beta plaques and neurofibrillary tangles, and move into example pharmacokinetic modeling of PIB, when it was a novel radial tracer, and then move on to tau, talking some about the first generation fluorotaucipir AV1451 or T807, now referred to as fluorotaucipir, and also a second generation MK6240 tau radial tracer. And then I'll end with efforts to gain more than one outcome from a dynamic PET study using these radio tracers that are not only do they provide measures of amyloid and tau deposition, but can they also, looking at the early kinetics, provide surrogate measures of relative perfusion? And then I'll point to future directions. So moving on, I'd just like to start with the traditional description of the amyloid cascade hypothesis in terms of how these proteins accumulate within the cytoplasm relative to the inside of the cell in the extracellular space. There are amyloid precursor protein spans from extracellular into the cytoplasm, but it's sequentially cleaved by beta secretase and gamma secretase enzymes, leaving fragments of 40 to 42 amino acids long. They can, if not cleared appropriately in a healthy way, they can begin to form oligomers, and that creates a toxic environment. And this hypothesis postulates that it is that toxic environment that leads to the tau hyperphosphorylation and cell death. And there are examples that support this hypothesis, including chromosomal mutations that lead to early onset Alzheimer's disease, and also chromosomal aberrations or abnormalities in Down syndrome that lead to the accumulation of amyloid and tau. So moving along, I want to just say a little something about tau now. It's a protein rich in neuronal microtubules, and it's normally there, and it stabilizes the microtubules, and they're key in axon protein transport in the neuron. But under toxic conditions or in disease, the tau subunits fall apart and they become aggregated in a similar manner, and it's aggregated or paired helical filament tau that we image with the PET. So just a little something about Alzheimer's disease. A major risk factor for late onset or sporadic Alzheimer's disease is age. In the 70 to 80 years of age, there's increasing risk. I've talked about amyloid and tau, but there's a precondition or a prodromal syndrome called mild cognitive impairment. It's really the type where you have a memory. There are different types of mild cognitive impairment, but if you have a memory deficit or amnestic MCI, that is considered prodromal in many cases to the development of Alzheimer's disease. So just moving on, I'd like to show this slide from the Pittsburgh group in terms of the problem and what we're actually dealing with. These are postmortem tissue samples. The left is an Alzheimer's disease brain, and the right is an age-matched elderly control. And you can see the abundance of neuropathological protein accumulations, where the tau-containing neurofibrillary tangles are shown here, the amyloid beta plaques there, and there's actually cerebrovascular amyloid that also accumulates in the vasculature. And this image was obtained using a highly fluorescent Congo red derivative used in neuropathology that stains the amyloid or beta pleated sheets deposit. But you can see the remarkable contrast between AD and control brain. And the challenge is to really have a radio tracer that selectively identifies amyloid beta plaques, selectively identifies tau amid the high concentrations that can be present of both. Now, just moving on to the sequential evaluation of PIB. This is many years ago now, and some of you have seen these slides, but it does still tell a story nonetheless. I wanted to start first with talking about in vitro receptor protein target parameters, just to define terms. The concentration of protein sites or receptors, in this case, they're protein sites, is termed Bmax, a nanomolar concentration. And there's also a parameter which we refer to as the equilibrium dissociation rate constant, which is associated with the binding process. The reciprocal of that is how tightly the radioligand binds to the protein target, or the affinity of the ligand for the receptor or protein. And we refer often to a binding potential that's the ratio of these two parameters. And just for a graphical depiction, the KD is a concentration that results in 50% of these protein targets occupied. It's the concentration that occupies 50%. And just in terms of tracer, when we deal with tracer levels where there's very little unlabeled radioligand present, and that's the concentration conditions that we often perform these studies, we describe the relationship between what's bound and free in terms of the Michaelis-Menten equation. And this nonlinearity primarily results because of an upper limit of the concentration of the protein. This produces a capacity limited situation. And when the amount of drug or radio tracer that is free is much, much less than that equilibrium dissociation rate constant, the bound over free is actually related to Bmax over KD. But that's just detail to help you understand these parameters that we try to then measure in vivo. I'd like to move now to the steps taken at Pittsburgh for the preclinical development of PIB. And again, why some of these, the pharmacology is important in understanding the dynamic PET signal that you model moving forward. This is an example, again, of postmortem tissue homogeneous binding assays, where we're able to incubate the tissue with the radiotracer according to specific protocols, and then later look at how much radiotracer was specifically or non-specifically bound to that tissue sample. And here again is bound over free versus bound, and that's referred to as a scattered plot. And this would be an example of a very high affinity binding site measured in AD gray matter, where the slope here is quite large, reflecting a very high, highly tightly bound radiotracer. But then if you look at AD white matter and control gray matter, there's very little evidence. It's just evidence of almost a zero slope and non-specific binding. And these were early studies, and you can see that refers to a KD of 4.4 nanomolar. The lower the KD, the higher the affinity, and very low affinity for the control gray matters, indicating specificity of the compound for AD. And then looking at brain homogeneous studies and trying to think forward about what might be an appropriate reference region. This is an example. This is a precursor of PIB. These are tritiated BTA1 studies. But when comparing elderly controls to AD tissue to people who had dementia, but not of the Alzheimer's type, you could see that really the binding of this precursor to PIB was quite high in the frontal cortex, but low for the other groups, and then similar across the cerebellum, suggesting that that would be an appropriate reference region moving forward in vivo. So moving forward, many of you have heard about the very first proof of concept studies performed with colleagues at Uppsala in Sweden. And basically, this is a summary figure from that first paper. This is SUV, no modeling, but a control subject that exhibited normal FDG metabolism, and an AD subject that exhibited nearly twofold higher PIB uptake in frontal cortex relative to the control. And this same AD subject showed the standard FDG hypometabolism, temporal parietal hypometabolism. But what we also saw was that there was a substantial uptake in white matter that is non-specific based on a prior slide. And so we began with these images through the brain. And again, you can see the non-specific uptake in white matter, but the contrast between AD and controls, looking at uptake over 40 to 60 minutes. So the next thing was to perform fully quantitative studies. And that included MR imaging that hadn't been included, longer acquisition and arterial sampling. And then we included some of the MCI subjects. And what we really sought to do was first look at quantitation on a regional basis, moving to voxel basis, if we're able to really identify simplified measures that we think are robust. But the overall goal being to study and evaluate the in vivo kinetics, determine what is lost and what is gained in the simplification. And then we really need the simplest measure, minimally influenced by blood flow changes in nonspecific binding and bias. Is it stable over time? So I'm just going to talk about a group of PIB studies conducted early on. It'll be either all of the 24 or a subset of these subjects. It's an older ECAT HR plus scanner, 90-minute acquisition, injected dose of PIB, 14 millicuries, and 40 samples, arterial samples from metabolite corrected input across the time period. These are examples of what we saw early on. This is the unmetabolized PIB in blood, starting at after injection through to the 90 minutes. You can see that over 90% was metabolized in vivo during this time period, and the absolute necessity to correct for this when determining your arterial input functions. And these are the early times and the late times for control MCI and AD. But what was encouraging here is that the metabolism and the arterial input function were similar across groups. And that was encouraging for simplification. Another thing we saw that was encouraging is that the cerebellar time, cerebellar to plasma ratios plateaued and were similar across groups. This is an area of the posterior cingulate that would be rich in protein neuropathological amyloid beta plaques. You can see it's the increased binding over time in AD, and it starts to plateau between 40 and 50, around 50 minutes. MCIs appear to be mid-range between the controls, where the plateau began a bit earlier than in AD subjects. But evidence of a plateau for both nonspecific and specific binding regions was also encouraging for simplification. So what we proceeded to do was explore the tissue kinetics. We started with spectral analysis to examine the kinetic components of tissue or impulse response, not assuming any model configuration. And then based on those results, look at the compartmental modeling, where we obviously assume specific compartmental configuration, and then look at a method that linearized the compartmental, or that linearized the equations to allow for a linear solution, assuming steady state had been established or pseudo equilibrium was established after a certain period of time. And the measurements that we refer to are the distribution volume ratio, which is the total volume of distribution, including blood, relative to the non-displaceable that we estimate in the reference region. Again, the cerebellum was what we proceeded with, and the binding potential that I mentioned earlier. In our in vivo work, we can estimate it as DVR -1 when using the cerebellum as a reference. And it's proportional to B availability divided by KD. And the difference between BAVL and Bmax is in vivo, some of the proteins for receptor work, some of the proteins are occupied by endogenous ligands. So we always try to remind ourselves that for receptor work. Okay, this is just the basic equations that we are faced with. Sum of exponentials for spectral analysis. The two tissue compartment model is shown here. K1 and K2 refer to the transport of the radiotracer across clearance from plasma into brain and from brain back to plasma. There's a free compartment for the radioligand that also is connected to a non-specifically bound compartment. Oftentimes the PET signal really doesn't, the kinetics of the radiotracer and the PET signal do not allow these to be kinetically distinguished for most of the radiotracers that we deal with. So we combine them into one compartment. And then K3 and K4 refer to the rate of binding of the radiotracer to the protein and dissociation. And this is a volume of distribution, is a macro parameter made up or described in terms of K1 and K2, and the K5 and K6 are wrapped into this K2. And you're familiar with the linear graphical method, where the slope is the volume of distribution when the equations are linearized in this manner. Okay, I just wanted to talk very quickly. I apologize. I have a lot of slides here. Many of them have information that's more for reference, because I just there isn't enough time to really describe in detail. But the simplified reference tissue model is something that is often used in place of an arterial dynamic study when arterial blood is not available. The simplified reference tissue is one that I'll be referring to. And just in terms of it really, the displaceable signal is assumed to be represented by the reference tissue. There's a two-compartment model in the target and or reference tissues, but generally a one tissue compartment model is often assumed for the reference region and the target region, whole blood contribution to target and reference regions, and differences in non-specific binding between the target and reference regions. So these are the basic assumptions, but in reality, oftentimes these assumptions are not satisfied, which is the point I really wanted to make there. SUVR tissue ratios, we talk about these often. They're just late scan ratios of the radiotracer concentration. And as a surrogate measure of the radiotracer distribution volume, that's actually defined at equilibrium. The concentration in the tissue relative to the plasma. And so they're basic assumptions. This is probably the most often used outcome measure, but it assumes a steady state between radio concentration and specific binding region and reference regions exist, and radio tracer delivery into these reference and tissue regions are the same. Yes, assumptions are not strictly met for leading A-beta and tau imaging agents because of radio tracer clearance in the region of interest, reference tissue, and although tissue ratios may be fairly constant, plasma is also clearing. As you saw from the arterial input functions, the plasma is not constant. Over time, it also is clearing. And this is relevant also for longitudinal changes in target protein deposition and patients with cerebral atrophy can also influence the blood flow differences across repeated follow-ups. So these are just things to think about when you're using the SUVR ratios. And these are two references that really go into detail about the use of SUVR tissue ratios in non-equilibrium dynamic conditions. So in terms of what we found with the spectral analysis, this is the way we visualize this data. The control is the posterior, saying that this is the region that in Alzheimer's disease, there was significant binding, but the posterior cingulate for the control overlapped with the cerebellum of the control and the cerebellum of the AD subject, with a very noticeable difference in the impulse response function for the AD, consistent with greater magnitude and slower clearance in this region of interest. But it also indicated that as many as 4 and as few as 2 components were observed, but most often they were 2 intermediate components. This could partly reflect the fact that we were imaging with a HR+, and there's some blurring of gray and white matter. And as you saw, there was quite a white matter signal. And then there's also noise that may sometimes give rise to a detectable component. But the 2 intermediate components were very consistently observed. Okay, this is just an example of initial modeling in the cerebellum using a 2-parameter model, and clearly the data were not well described. Using a 3-parameter model, the delivery characteristics were still underdescribed. But a 4-parameter model gave the best fit in terms of the IKAK criteria as well, which is a parameter that many of you are familiar with, which relates the goodness of fit for nested models. And the standard errors of the kinetic parameters were also very low, giving us confidence that we were actually fitting the curve in a robust manner. So this is just another example of outcome measures that we obtained for a 2-tissue 4-parameter model, 5 controls and 380 subjects. K1, K2, but in the control, we referred to K5 and K6 as a non-specific component, rather because K3 and K4 was reserved for specific binding of the radio tracer to the, and the control values were fairly similar, whether in cerebellum or posterior cingulate, but you can definitely see the difference in the ASPEC, particularly with respect to the non-specific component relative to K5 relative to K3. That was nearly 4 times greater, consistent with a significant specific binding component. And we did try to fix the K5 and K6 parameters, but when we did that, it led to unstable results and sometimes negative values, meaning that the data just couldn't support even parameters even when constraining either K1 over K2 or the K5 over K6 ratio. And then the next thing is, what about how did the simplified method relate to the compartmental method? And this is just an example of the posterior cingulate and parietal that would have significant protein concentrations in AD, and good agreement was found with some bias relative to, for the Logan method relative to the 2T compartment. And we know the Logan is susceptible to noise-induced bias. So it's important to always keep an eye on how robust the Logan is, particularly on a voxel basis. And then I'm just going to show a graph of how relating the outcomes across these various methods that I've described already. We did look at a carotid input function. We didn't do it in the way it should have been done, but we did determine carotid input function using data over 0 to, based on ROIs generated for data over 0 to 45 seconds when the carotid was most evident. And using a population average metabolite curve, we corrected the carotid radioactivity. And the average arterial and the average carotid had a very similar shape, but you can definitely see a difference in magnitude with an underestimation of the carotid and the need for a careful partial volume correction and motion correction when you're using the carotid radioactivity and trying to, as an image-derived input. But nonetheless, these are results across control and AD for the different methods. The standardized uptake ratio over 40-60 minutes did well at distinguishing AD and controls. All methods did, but the difficulty really becomes in the MCI group that we now could see we're not in the middle. Some were more control-like and really some were more AD-like. And that's just shown here how the range of uptake varies. You can have a normal control. These are people who are cognitively thinking normally, evidence of just a little bit of accumulation. And these are people who have cognitive deficits, but some may not be of the Alzheimer's type, and some can have as much accumulation as AD. So I just wanted to say a little bit more about the SUVR. This is an example of computer simulations that we perform using average parameters. And we looked at the DVR with blood. These are the DVR values that we calculated for an 80 subject and a control. And then we computed the SUVR over the time period. And you can see how the SUVR in this case overestimated the DVR. And in this, for a control, it's much less so. And what really, this is an important thing because many studies are really moving over the last, I'd say, 5 to 10 years. There's been an effort to really go to the cognitively normal. And so I think that's because of the lower bias in these subjects, when there isn't high binding, the data has really provided a lot of insight into the in vivo progression of this disease. So the 40 to 60 minute window was probably the best to choose here, although you have to be very careful where you are in high binders and that you put them, if they come in for an uptake period, you really need to get them in the scanner at the right period of time over time. So I just wanted to say something very quickly about bolus and infusion imaging. These are some slides provided by Dr. Koeppe. And really, you can offset the clearance using an infusion protocol where you prime with a bolus and then you follow with an infusion. And this is an example of graphs that he shared for PIB, where this is the bolus PIB. And this is what you can accomplish as long as the kinetics across the different regions are similar. And in the case of PIB, the kinetics are similar across these regions, and they were able to achieve a fairly constant concentration after later times, across all of these regions. And you can see the cerebellum's just a little bit later, but it also plateaus. And these are the tissue, this is the PIB concentration, and this is the tissue concentration ratios. And you can see how they really do. This is bolus, does not, I showed you what bolus looks like, but then you can really see the bolus plus infusion, how it flattens out. So this is another method that can be used, and this can allow for short-term imaging when you really, despite the binding characteristics, you really can offset that with the infusion protocol. And then he also shared this slide showing the differences in DVR estimates as you go from bolus with the tissue bolus Logan into the equilibrium and non-invasive Logan methods. And you can see some evidence of the bias, particularly between the bolus method and the reference Logan method. Okay, so what about tau? I'm going to go a little quickly here. I'm a little behind time, but what about tau? I'll talk something about the emergence of tau, example kinetic evaluations. The evaluations for tau came a little bit later. It was beginning to be very widely used, and the modeling came a little later, and there was some cost to that. This is just a summary of some of the in vivo studies that took place between 2016 and 2019 for different tau radio tracers. And this is an example of a subject scanned with 3 radio tracers, which is fluorothalsapyr, THK 5351, and then a newer generation in K6240. I'm going to say something about off-target binding was a concern with these radioligands, and I'll say more about the putamen, particularly for fluorothalsapyr. But in postmortem studies really helped to further understand the nature of this off-target binding. But what was also revealed is that for THK 5351, the sizable fraction of that binding in brain was attributable to binding to the monoamine oxidase B and not to the protein, not to tau. And so it's very important to do some of these evaluations as early on as possible before widespread use, and this is no longer used. Just showing an example of target binding for FLIR and how it also has an age related component to it. Again, that putamen is popping up where it's not really as evident relatively in young controls. This is an MCI. We're really starting to see the tau accumulation in mesotemporal cortex and cortical areas. And then AD, this is just widespread binding of the tau. And you can see also the uptake in putamen. Moving along again, we look at our time activity curves. And this is just showing for the putamen that off-target binding, how it looks relative to other regions in a healthy control. But in an AD subject, you can see that the putamen signal is quite different than a tau-rich cortex area. And relative to the cerebellum, which is shown in black, that has the greatest clearance. And then if just looking at the SUVR normalized with the denominator, again, relative to cerebellum, again, for healthy controls, the plateau occurs earlier, but the imaging for some of these traces really require longer time periods. And that's really evident in the AD subject, where for the temporal cortex, the binding is still increasing. Again, the putamen and the off-target binding is shown here, but this is a little bit more of a challenging situation because often the subjects have to take a break. And the folks at Lilly's suggested that there be a correction applied for differences in delay from the injection to the scan time. As I mentioned, it's really important to put them in the scanner at the appropriate time, but sometimes in clinical studies, that's not possible, so they offered an equation to correct. And moving forward, I'm not going to go through these in great detail, but this is an example of the type of analysis that was done eventually for fluorotaucipir. Again, we started including very young people, and that's important. Elderly, 59, and AD. Again, you can see there was more than one break in this study, but now we're up to 210 minutes. This is an example, 2-compartment fits, the Logan graphical, linear regression, the simplified reference tissue fits, and you can see differences in fitting of the dynamic data between the two tissue and the reference tissue. And then this is the non-invasive Logan fit. And so the linearity was established, but you can see it's more of a restricted dynamic range for the non-invasive Logan. Okay, moving along, we cut the time period down, and I think that they began then to look at 80 to 100 minutes versus 110 to 130 minutes, particularly for the SUVR. And there is an obvious bias even between the SUVR methods, but high correlation. Okay, so the fluorotaucipir imaging really indicated that it's the 80 to 100 minute interval was a good choice for late scan cross control and AD subjects, but still concerned about off target and the lack of SUVR plateau in high binding regions. Despite those problems, this is an example of what people were seeing at the time. This is a control with no cognitive deficits at all, and it ranges all the way across the AD spectrum to someone with advanced Alzheimer's disease. And you can see that the uptake is correlates with the clinical progression of the disease. And it also followed the Sprach staging that neuropathologists use when they evaluate postmortem samples. So there was still a need to reduce off-target binding, to improve the in vivo kinetics, to ensure stable quantification of tau load, even though people were moving more toward low binders and earliest detection of disease. But greater signal to noise was needed. So enter MK6240. There were several early papers on this. There's clearly a push for reference tissue. What should the gold standard be? Should it really be arterial anymore? But there was a combination of early studies that indicated 70 to 90 minute SUVR, 60 to 90 minute SUVR. Test retest data was being presented, but what was becoming evident was that there was, although you don't see the putamen uptake off-target signal at these time periods, there was some halo of radioactivity that some subjects exhibited and others did not. And this is shown in the paper, the MGH paper by Nicholas Gell and Marc Normandin and others. And this is an example of subjects, control subjects and several MCI-AD subjects. You can see in some of the controls, you have this outer ring of intense radioactivity concentration, and it's even apparent in some of the MCI subjects. And they presented pharmacokinetic modeling strategies. This is a slide from Bob Koeppe that shows, I think this was a Pointer study, another example of this, where this is an amyloid, the fluor beta bin amyloid scan, but overlaid is the MK6240 tau uptake, that extra cerebral uptake. And this was as high as an SUVR of 14. So this is what's in the consideration is, how does this impact quantification of the reference region and cortical regions for this radio tracer, despite the lack of putamen non-target binding? What do we do? So it appears that the MK binds to neuromelanin and melanin-containing cells, but I don't think that we still fully understand the true nature of that extracellular component. And one of the things that we took a look at, again, was spectral analysis, just to decompose and see how the cerebellar gray matter would compare to this extracerebral signal. And there was a significantly larger trapping component. This could be noise, or it could just be maybe contamination by white matter. But these studies were done with current day, and really the GE digital scanner, and we really found that there was this significant trapping component. The frontal cortex and cortical regions, the spectral analysis really was more like the cerebellar gray matter in terms of a very small trapping component. And then a slower and a faster reversible component. But it was the magnitude of the trapping relative to the faster reversible that was really notable for the extra cerebral signal. And that was found to be similar between diagnostic groups with similar magnitude, but the extracellular change in SUV from 6 months to 1 year was much greater. And this component was found in about 50% of the subjects. Now I'm going to move along again. I'm sorry I'm going over here. I just want to say something about relative radioligand delivery. Blood flow, blood-brain barrier kinetics. We look at delivery in terms of its flow and extraction across a blood-brain barrier. Reference tissue based models estimate R1, where R1 is the K1 of the target over the K1 of the reference. And we were interested in relating radioligand R1 to the gold standard O15 water R1. And if you don't have a water R1, how might you go about doing this? This is just a unit capillary model that we're familiar with. Arterial blood enters with arterial concentration, exits with venous concentration, and the venous concentration difference is what is used to determine a net flux. We use a Rankin-Krohn model to account for the fact that tracer diffuses, although well mixed in an extravascular space, there is a concentration variation along the vessel. And that's described in terms of the extraction component as 1 minus e to the permeability surface area divided by the flow. And when we estimate our K1, and I apologize for not going through this in detail, but again, some of these are for reference. This K1 or delivery component is really flow times extraction. And that's what we are measuring. This is an example of study that we did with PIB relative to the simplified reference tissue method R1 measurement. And we just basically found very similar distribution of the R1, whether it's the ratio determined using water or the ratio determined using PIB, particularly in cortical areas. Some areas like white matter, it just doesn't perform because of the nature of the tissue. It just wasn't performing strongly. And I just wanted to make a mention of the Amsterdam group. They looked at test retest variability of R1, and they found, again, moderately high correlations. And they were concerned about the extraction, variation in extraction. So they looked at a group of controls, MCI-AD, and their relative R1 measurements across diagnostic groups were clearly the R1 was much lower in AD, and that can be because of vascular effects. It can be from increased atrophy relative to these groups, but it was very reproducible upon retest. And that was true across most regions. This is the level of the variation that they found in their test retest data. So for PIB, R1 was pretty robust. Then Dr. Gell and Dr. Normandin and Gordon Center MGH colleagues in a group effort, we looked at MK6240 and PIB, and he showed that PIB R1 relative to the MK6240 R1 were very highly correlated. This is across regions, but still very highly correlated with low slope bias, and also showed beautiful parametric images of MKR1, PIB-R1 for a control subject, an 80 subject with evidence of lower relative perfusion in areas of high tau uptake for the AD subject. So if you don't have O15 water, one option might be, if you do have some dynamic PIB data, to relate the R1 for new radiotracer relative to PIB. And then this is, I apologize for being so busy, but this group, we are also working on finalizing relationships between O15 water. And I'll just move us here. But clearly there's a bias when looking at this just K1 of MK relative to water. And that is basically mitigated by comparing the R1 when taking the relative ratio for MK relative to water. And so we think that this is just further verification that the early MK data can be used as a surrogate measure of perfusion. And then I wanted to end with some future directions. You all are very familiar with the new just high sensitivity total body scanner, but it really does hold promise for what we might be able to do in terms of modeling, not just in the brain, but throughout the body. The type we can use and the whole body kinetics that we might be able to model moving forward. So this is an area of great promise where the blood, the ventricle radioactivity concentration is in the field of view. And that's just the beginning. And the high sensitivity might really allow us to use more sophisticated compartmental or differential equations. Okay, and I just wanted to end with something going on at the Martino Center by Dr. Katana in terms of a development of a human dynamic neurochemical connectome scanner. And this is also something to point to for the future as it'll combine PET and MR at 7T for human imaging. And I look forward to that as a future direction to maximize sensitivity by addressing geometric efficiency, detection efficiency, improving across many parameters to really improve sensitivity by using a scanner configuration of this type. So that's just an example of another total, in addition to total body, there's a tomograph that's being developed. And that's all for today. Thank you very much.
Thank you, Julie, for that wonderful talk. We'll now move to the panel discussion and Q&A portion of the session. To everybody in the audience, if you have any questions that you'd like to ask or comments that you'd like to voice, please indicate so in the chat or by raising your hand in the Zoom reactions panel. While the audience gathers their thoughts, I'll take a moment to introduce our panelist, Dr. Robert Koeppe. He's a professor of radiology at the University of Michigan and the director of the PET physics section in the Division of Nuclear Medicine. His expertise is in the quantitative analysis of PET data, and he's been an integral part of many large multi-center imaging trials involving PET tracers looking at glucose metabolism, amyloid, and tau in the study of aging and dementia. He received his bachelor's degree in physics and a master's degree in nuclear engineering at the University of Missouri-Columbia, and he earned his master's and PhD in medical physics at the University of Wisconsin at Madison. He's been at the University of Michigan since 1985 and has received numerous honors, including the Hal Anger Lectureship Award from the Society of Nuclear Medicine and Molecular Imaging. So by all accounts, extremely knowledgeable, experienced, and the ideal person to offer thoughts on the work that Julie just presented. Bob, to start off the panel discussion, would you like to share some initial comments on Julie's presentation or ideas on topics covered?
Sure. Two things. Just want to kind of reiterate the fact that I think that Pittsburgh's work, Julie primarily responsible for the kinetic analyses, is they really did this right. They took a tracer, they started with in vitro studies, moved to in vivo, they did the full kinetic modeling, they looked at everything they could to get simplifications, and now PIB and other amyloid studies which behave similarly are in countless multi-center trials. So if you're developing a new tracer, go back to these slides, rewatch the talk, and go back to all the slides on the PIB. And this is how you do a proper validation of a new tracer. The other thing, we've also worked and I think I can share screen just to show one slide. We've also looked at the K1 measures. And so this is a different tracer, not an amyloid tracer. It's dihydrotetrabenazine, but just to show how similar the K1 measure is for many, many tracers to the glucose metabolism, which of course a lot of people do that. So here in frontal temporal dementia, AD, Lewy body dementia, and normal controls, you can see how very similar the K1 map is to the FDG. So if you're willing to do a dynamic study, you can get almost as good quality as an FDG scan, so 2 for 1. So, and the final thing is, I think Julie also was correct that with the success of amyloid, I think a few of the initial validation steps for tau tracers were missed, kind of caught up on that, but I think there were more problems getting started. Plus the tracers aren't quite so good for tau as they are for amyloid, and we may get to discuss that a little bit. But with that, thank you, Julie. That was great talk. And let's go to other discussion items and questions.
Sure. I see a hand raised, Mary Ellen Karan. Would you like to offer your
Hi.
question?
Yeah, thanks. Thanks for this talk. I'm coming from Vanderbilt. I'm a nuclear medicine physician in the Alzheimer's Center. We are getting dynamic PIB and MK6240, but we don't have ARTLine data. And I was just curious about the R1 that you're able to derive. Do you need ARTLine data to be able to derive that, the relative radioligand delivery R1 that you were talking about, Julie?
No, the R1, and I apologize for not being clear on this, but that is a parameter that one can estimate from the simplified reference tissue model. It's a parameter available through that, and yeah, you use the reference region as an input for that type of model.
You do need to
great,
scan.
thank you.
You do need to scan with injection. You can't do just light scanning to get the R1.
Yeah, we have 0 to 70 for PIB.
Yeah.
Okay. Great, thank you.
Okay, we have a couple of questions that have come in the chat. Chris, would you like to read your comment, or I can do it for you? You'll do a better job. Okay. I'm not hearing anything, so I'll read from the chat. Okay, Chris doesn't have a mic nearby. So Chris's comment reads, "Dear Julie, many thanks for the amazing overview. I had a question regarding the data presented regarding tau. Using the T-break protocols of 210 minutes for tau imaging, were measurements taken to ensure the same brain relocation in the scanner after the break as the repositioning of the subject in the scanner alters the tissue constituents of the voxels in the images, and hence the quantification." And there are some follow-ups that maybe we can address afterwards.
So I think these are very important points. I think sometimes if you're performing say, a PET-MR simultaneous study, you can do an MR scan, multiple MR scans that allow you to then at least register the MR scans together, because you can do this MR imaging during the PET scan. And the spectral analysis data that I was showing definitely was from that type of data. But your point is well taken. It's very important to really account for motion as well as you can, which still doesn't necessarily solve the problems. We have attenuation issues as well. But I think it's a matter of trying to be as careful as possible when you are performing these studies watching it. My had a way to go. I don't know if others, if Mark or Bob have comments on that.
I think you do have to do a CT scan before each session. I don't think you can rely on getting the subject back in the same position. So one of the larger problems, I think correction for motion of the PET itself works very well, and you can do that across. But if the CT and the PET don't align, then you're going to have attenuation correction artifacts. So if there's 2 or 3 time periods of acquisition, you'll need 2 or 3 CT scans.
Yes, or MRs.
Or MRs.
Yeah. And one of Chris's follow-on comments is that these concerns become all the more dramatic as the resolution of scanners improves. Maybe that's something that we can hold off on and talk about a little bit more in depth a little later. We have another question from Terry. Terry, do you have audio available?
Yes. Julie, thank you very much for your talk. With the new sensitive machines, factor 10, will it be possible to follow the clearance of the armor after you tagged it, then to see it being taken away through the CSF, which is the big, as you know, the action now, these therapeutic agents trying to clear the amyloid. Do you think it'd be possible? It won't have high resolution necessary, but over some hours to get a rate of clearance of the amyloid from the brain?
Well, that would definitely be an interesting thing to look into. I really don't. I don't have an answer, but I think that hopefully we will have the opportunity. And I think this is where pairing it with some of the MR based outcomes might really, in terms of maybe in terms of pulsatile motion or things like this, it might, it'll be an interesting area to examine. I don't know if you have experience doing this yet, but it's certainly a very interesting and very important area is the lymphatics or the clearance of the amyloid in the presence of the therapeutics over time. Is that what you meant?
Yes.
Yeah, that would be something very interesting to explore, and I hope we can do that.
Okay. Well, Bob, would you like to read your comment? Oh, sorry, Bob. Did you want to? Go ahead, Bob.
I don't think I have anything additional to say on that topic.
Okay. Bob, did you want to comment on R1 and K1?
Sure. Yeah, because Julian and Bob mentioned that both R1 and K1, and so for brain dedicated imaging, perhaps R1 is the parameter that we can get, but if we can also get a good image-derived input function or arterial blood input function, then we could also estimate the K1 directly. Just wondering, Julie and Bob could comment on the potential advantage and the disadvantage of using each of these 2 parameters.
Okay.
I guess,
go ahead, Bob.
I was just going to say it's really, to me, the issue of, are you going to do arterial sampling or not? So in most cases, we're not. So having a R1 measure, which does again require imaging from injection, but it's much more applicable to a lot of studies. And as long as the extraction fraction, so Julie mentioned that K1 is flow times extraction fraction. And so you hope that extraction is high enough that the R1 or K1 gives you an image that looks like flow, and flow looks like FDG in many neurodegenerative disorders. So I think the main limiting factor for both R1 and K1 is whether you have a tracer that has a reasonably high extraction fraction. You know, if it's 5% extracted, it's probably not going to work very well. If it's 50% or more, it's going to work very well. And somewhere in between, it transitions from quite good to not so good.
And Mark, if I'm not mistaken, that was one of the thoughts that you all had initially about MK6240.
Yeah, so
It's higher
what it
extraction.
kind of provoked us to into this investigation was, you know, the observation that when we did blood-based modeling, we were getting, you know, moderately high K1 values, which implies pretty good extraction. And so from there, it led to looking into R1 as a surrogate perfusion index. Julie, I think you also showed some data, I believe also with MK comparing to water, where even when the K1 values are not perfectly correlated, so in other words, the extraction of MK is actually not high enough to get absolute quantification, the R1 values were still in strong agreement. Do you want to elaborate on that a little bit?
Well, I would just say that I think there's a couple of things with the ratios. I mean, we saw that also with PIB. In some areas, the performance in other areas, that's not the case, because the extraction characteristics in the cerebellum may be very different than white matter, for example. But for PIB, we found pretty much the same type of thing. And I think that it's just that the extraction is good enough for R1, but just not good enough for the K1. And then K1 is a parameter that obviously depends on how the radio tracer is injected. If it's, you know, if it's injected more rapidly or more slowly, but just I think the R1 helps to also just kind of remove some of the variation in how in the injection as well, if that makes sense.
Yeah. Okay.
Can I ask one question?
Please.
Hi, Julie. Yeah.
Hi.
Thank you for the amazing talk. And I have one question about the PIB. It seems like the radio metabolite of the PIB is very fast, and the radio metabolite its effect can be ruled out by kinetic modeling. This is one question, and the second is if the radio metabolites happen very fast, and you mentioned you use bonus injection and bonus and perfusion, and would these both injection method would affect by the radio metabolite?
Well, I would say that if you're doing modeling with arterial sampling, if you're able to correct the metabolites well, then it's not as much of a problem as when you're not. And that's what you can avoid with the sort of the trade-offs or the things that you consider when you think about whether should I model K1 ratio or should I. It's not easy measuring radio labeled metabolites, and it's time consuming, and you really need expertise to do it. So I would say that if you can do that well, then it's accounted for in the kinetic modeling. But I forgot the second question you had, and then I'm going to also ask my moderator and panelists to make sure that I understood all aspects of this question. But what was the second question you asked?
The second question is about the bonus injection and perfusion.
Oh,
Yeah.
Oh, okay. So generally speaking, it's really the early data that I think is most important here for the relative delivery measures. We've also looked at Marc and our groups together have looked at just looking at maybe 3 minutes of data. And for some of these radio tracers, including MK6240, that actually, and PIB, that can also, that the ratio of the early, just the early data can be useful as well. Marc, did I describe that? Is that your experience?
Yes. One thing I wasn't, I just wanted to clarify about the question is the context, and I guess the time scale in particular. Were you asking about this in the context of measuring a perfusion metric, or was this towards full kinetic modeling of 90 minutes of data, or all of the above?
I'm concerned if the radio metabolite happen, and if you slowly infusion the tracer, and then maybe the SUV will be affected by the radio metabolites. So that's so it will appear, yeah, the bonus.
There's a basic assumption here, maybe. The basic assumption is that radio-labeled metabolites do not enter the brain. And I think you know that, but I'm just saying that because I want to make sure I understand the question. But I think if you have a novel radio tracer, expect that there are radio-labeled metabolites, and sometimes you can see that. You can see it can be suggested by the shape of the time activity curve. If you have a reference region that continues to maybe increase at late times or things like that, then that's a basis for concern if you know you have a radio tracer that should be clearing. But generally speaking, if I understand, you should be able to correct for radio-labeled metabolites. And I am happy to talk to you more about this if you want to after the talk too.
Yep, thank you, yeah.
And I would agree with everything that Julie said. You know, the first thing that always comes to mind for me is do the metabolites, the radio-labeled metabolites enter the brain? If they do, that's a big red flag from the start. If they don't enter the brain, then I guess the question is, do they exist at all? Preferably they don't, then there's no corrections needed to your input function. If they do exist, is it possible to use a population average? Those things need to be validated. I guess the other question about the bolus infusion is that there are some instances where the metabolite levels drop off so fast that getting accurate metabolite data is challenging. We had some difficulties of that nature with MK6240, for example. And although we didn't perform what I'm about to suggest, it is theoretically possible to use something like a bolus infusion protocol to, you might say, slow down your metabolite parent fraction curve over time. You're not actually slowing metabolism in a physiological sense, but you're protracting the input function. So essentially you're convolving the impulse response function that you get with a bolus injection with something of longer duration, and that gives you you know, a longer period of time to measure the metabolites. And if you reach an equilibrium state, you don't need to do any modeling at all.
And we actually do bolus plus infusion for quite a few tracers. We've not as much recently, but we, 10, 20 years ago, we did a lot with arterial sampling with metabolite corrections, and most tracers as Marc just mentioned, you not only get the brain tissue, but the blood and the metabolite corrected plasma to a uniform concentration. And then I think it was Julie mentioned that besides an R1 measure, for some of the tracers, the voxel-wise estimate of R1 is kind of noisy. You have fitting errors. So we have often gone to, summing the first 3 to 6 minutes and using that as a surrogate of R1. It's the early uptake. It looks very much like flow. And you never have any fitting errors when you just sum the data up.
So I had a question. We've sort of indirectly worked around this point, but I'll just kind of try to articulate it. What are the characteristics that make for a good or improved radio tracer? And in the context of some of the radio tracers that you talked about, Julie, some of them work very well. Some of them work well under certain instances, but they have limitations. Is there room for improvement, or is what we have good enough?
Well, I would say robust blood-brain barrier clearance is one. You don't want it to be shuttled back out by a efflux pump or something like that. So you definitely, is there room for improvement? Reducing non-specific binding is really important, but we know in our experience, sometimes a little bit of non-specific binding is actually helpful because there was a radio tracer a long time ago that had very low non-specific binding, and we were often faced with the impact of scatter correction when you really get to low signal and the noise in terms of really quantifying reference regions, which are with us all. But when you really, so I think it's a balance, non-specific binding. It depends on action, but you would like to have a solid measure of your targeted binding parameter within a feasible time frame. And I think those are just, when it's the equilibrium between binding and dissociation, and you just have to balance those things. But there are papers, I know Chet, Victor Pike, and several others have papers that actually define the ideal parameters, so to speak, for the receptor imaging and protein targeted imaging, and affinity, specificity, selectivity, all of those things are important. So I have a slide to show, but I think selectivity is very important. All of those things, it just, we can always try to do better. I don't know. What do you think about that?
I'll punt that over to Bob.
Okay. Can we do better, Bob? Oh, the tomographs.
We can do better for several reasons, but before we get to that, I want to just second what you said about you need some activity in the reference region. I was thinking about questions to bring up, and one of them is anybody can type it in the chat or make a guess, but what would people think would be the optimal ratio of target to reference binding? 1-to-1, that doesn't work. 100-to-1 is too high. So in my experience, exactly what Julie was saying, because you need to just think of taking a ratio, numerator divided by denominator, And if you have a scatter correction, which is subtracting 30% of the counts off, if you have 100-to-1 ratio and you miss that denominator of 1 by, a 0.5% out of the 100, that's a 50% change in the ratio. So that's way too high a ratio. So the question is, where do we think the optimal target to reference region ratio is for PET right now? People can throw that up as their guess or opinion in the chat. Julie or Mark, do you wanna, or anybody venture? I sort of have
Are we restricting ourselves to short-lived isotopes?
Well, C11 or F18
Okay,
was what I was thinking.
I've got a number in mind.
I'm going to. I think
You want to go first or second, Mark?
I'll go first. I'm going to say 5. I have 5
I was
fingers.
going to say 5 too.
I was going to say up to 10. 5 is good. By the time you get to 15, I think you've passed the optimal point where, if it's even 10-to-1, if you have a few percent error in the scatter, that can start to be a problem. So if you get a 30-to-1 ratio in a tracer, that's probably not the best tracer for what your target is.
And this is an example of a PK course question that Bob might pose. And if you're aware of the PK course, and you're interested, I would encourage you to take it. It is attached to several meetings, like S&M neuroreceptor mapping. But I couldn't remember exactly if it was 10, Bob, or 5. But do you think 5 to 10 is a reasonable range?
I think so, yeah. Even tracers, people are used to tau and amyloid, and seldom do you have SUVRs of higher than 5.
So flumazenil might be 5, right?
Flumazenil, yeah, 5 or 6,
Yeah.
and it's a good tracer. MK is, you can see 10 fairly regularly. In the PK course, I actually asked the question, if you're doing kinetic modeling, what's the ratio of K3 to K2 that's optimal? Which is sort of the same question, but for kinetic studies rather than static studies.
Quick question, if I may add here too, because this scatter was picked on, but how about randoms or wrong attenuation correction? Because the amount of problems you can have in PET is just not on the scatter. Could be, for example, randoms going on quite depending on the distribution. But why is it particularly scatter from your experience?
Well, that's a good point. I do think scatter's the most important of the 3, that randoms are probably more accurately corrected. Scatter is because in my opinion, if 30% of your counts, you might be subtracting off a fairly large fraction of the signal. So I'm just going to make up a number, but say the raw signal is 50, and if 30 or 40 of that is scatter, and you make an error of instead of 30 subtract 35, that's a 25% error in the denominator. So with attenuation correction, it's a little more predictable correction. It's fairly large, but you're not going to make a 20% error in the attenuation correction. Randoms I haven't thought about, so yeah, that will contribute as well. But I'm guessing that the count rates are, in the same ballpark for these tracers, so the randoms level won't vary a lot from scan to scan.
But we do have problems with motion,
Yeah.
during the PET scan in terms of the attenuation.
Yeah, absolutely.
This really
so-
can be problematic.
Yeah, so in these multi-center trials, I've gone to saying that the most important thing is to do whatever you can to prevent motion between the CT and the PET. I think some sites make a mistake and they do the CT and then they tell the patient to get comfortable because they have to hold still for a long time. That's not the time to do it. You tell them before the CT to get comfortable and hold still, and tell the subjects, the bed is going to move from the CT to the PET. It's important to remain still. One more comment on that, at least for Siemens, they provide the ability to translate the PET relative to the CT. They don't provide rotations at this point. So that can save scans that otherwise would be lost. I don't know about GE and Philips. And I think, there's new software coming out of the scanners that will do better at having CTs match PET with post-processing algorithms. I don't know, Mark or Julie, I don't know that much about details of those, but that's a big issue for the future, I think.
Yeah.
There's also, because the motion, from my experience, is that the motion is one thing, as you mentioned, is it's the correspondence of the mu map or the CT, however you get the mu map from, with the PET data at any given time.
Yes.
And another thing is then also to follow that with your sampling masks. So for example, if you get cerebellum as your reference region, that if that's not sampled correctly, that also, so the motion can kill you multiple times over the extracting of the signal and getting the right signal. Do you have any comment on, what is the worst?
In my experience, you can very nicely correct for motion within the PET. And it's the CT to PET mismatch that causes the problems, that the mu map from the CT or for a map from an MR scan. So what I was mentioning is that if there's motion in the PET, then you may need to apply different attenuation correction maps to different frames. And the need to have moderately short scan frame times will mean that you don't have blurring within a frame. Sometimes the person moves so much within a 5-minute period that you have to throw a frame out. But most of the time, even with the newer higher resolution scanners, I'm fairly impressed that after motion correction in the PET itself, the images still look very sharp. But one final state, to repeat again, the attenuation correction map needs to be, in a sense, registered to the PET for each frame. We need better ways of doing that for the future.
I see that we have actually just run slightly over time. I hate to cut off a good discussion, but in the interest of being punctual, I think we'll need to bring this session to a close. Thank you, Julie, for a great talk. Bob, for your insightful thoughts shared today. Bobo, thank you for organizing this session and leading the Open Kinetic Modeling Initiative. Thanks to all the members of the audience for your attention and interactive participation during today's session. Hope you have a wonderful rest of your day and hope to see you at the next quarterly webinar. Take care.
Thanks, Mark.
Mark,
Thanks.
thank you so much for moderating the whole session.
Sure thing.